All right, I think we've got everyone. Welcome back from lunch. I guess I'm just the after lunch person for your program one. We're going to be talking now about some more types of validation experiments. So this morning, Andy did a good job kind of touching the kind of practical applications of all these things that we're telling you. So this section is going to be on peptide stability. We mentioned that a little bit in passing. We're going to talk about peptide reproducibility. And we're going to talk about peptide repeatability. So it sounds like I just said the same thing 3 times, and you're kind of right, but they all have very distinct definitions in what we're going to be talking about. Before we step into validation experiments, I just wanted to give you a little bit more context about where we are as far as kind of like all of this information we have dumped on you. And we've just kind of like thrown you in the deep end of this extremely complex and very deep software structure. So I just wanted to take that step back and kind of review a lot of the things that you've learned. So with a targeted mass spectrometry experiment, researchers, we always start with some hypothesis. And that's true for SRM and MRM. Which is kind of the topic of this course, is SRM, MRM, and kind of PRM, so a targeted assay. We have had some people asking about DIA and DDA. You will be learning, I asked Olga yesterday, you're going to be learning more about DIA later, so all those questions, definitely keep them in mind, and they'll be gone over later in the course. With SRM or MRM, though, you have to know what you're looking for before you can look for it. So we went over a little bit yesterday how Skyline, if you had some protein of interest, you could go, for example, to Uniprot and say, I'm interested in vitamin D binding protein, and download the FASTA, and then we talked about, oh, there's different isoforms, how's that going to mess up my experiment? We went into Skyline and we played around with our different settings, and we said, let's say 1 miscleavage or no miscleavage. Let's say I want amino acids from length 2 to length 100 versus a length of 6 to a length of 25, and how that was affecting some of the different peptides that we might be able to target. Yesterday, Sue went over all the different peptide transition selection parameters that you'd have to think about if you were making one of these assays. We went over retention time yesterday. We went over collision energy yesterday. We played with spectral libraries, which you might generate from your own DDA, or you might go to some repository online and download someone else's spectral library. I had shown a couple people about method exporting. We're not talking about that a lot in this course. We're kind of taking for granted that you might know how to run your instrument. But if you go in Skyline and go File, Export, there's an option on the other side that says Method Export. There's also an option for Transition Export and Isolation Export, and all of these are tools to help you on your particular instrument that you have available. Acquisition, we've been talking mostly about triple quad. I personally run mostly on an Orbitrap now, so if I'm doing a targeted assay, I'm doing a PRM. Skyline's vendor neutral, so it's going to take any data from that and import it and treat it just the same as data from any other type of instrument. We haven't talked at all about processing, but I'm going to talk a little bit about how Skyline's processing this data. All that we know so far from this course is I set up a target list and I import raw data and something happens, and then I have chromatograms. There's a lot of pretty cool stuff that's going on under the hood when that little animated chromatogram is going across your screen. We've shown you some of the core analyses that Skyline does. These are all those different interactive panes that update whenever you click them or whenever you click a different peptide. So some of those core analyses are things like that retention time pane with all the comparison plots and the regression, the peak area comparison graphs, and we played with grouping by, and we played with normalizing areas. We haven't really talked about group comparisons yet. I think you're going to do that a little bit more later this week. But there's ways to do a group comparison. So if I have healthy and disease, I can do the statistical comparison of those two just within Skyline. Some of the additional analyses that you can do, we've also mentioned and then kind of immediately moved on to something else. But there's all kinds of external tools that you can install in Skyline for that vitamin D binding protein that we were talking about yesterday. There's an external tool called population variation. from PNNL that you can test any of the proteins in your Skyline document for variants that are prevalent in the population. It queries a database. So all of these are kind of wrapped up in the Skyline ecosystem. And I just wanted to applaud you. We're halfway through, a little over halfway through the course. You guys have already done everything on this chart. So if it feels like you've been a little lost, don't feel lost at all because you've actually learned a lot. So what I want you to take away from this lecture portion, before we go into the hands-on stuff, all I really want you to take away from this is to be able to think about validation in relation to whatever projects you're doing. And I'll get into this in a little bit about how different experiments might require different levels of validation. If you're an academic researcher and you're just kind of doing very preliminary analyses of proteomes, maybe you don't need to necessarily do every single stage of validation. But if you're a clinical mass spectrometrist and you're working with patient samples, maybe you really want to validate all of the targets in your assay. So I wanted to just start by reviewing some figures of merit. We had talked about setting up this kind of method development, right? And I just walked you through some of the tools that you now are equipped with for doing this in the Skyline ecosystem. So now oh yeah.
Is it online?
Yep, all of these slides are all in that Google Drive. All the slides from forever. They should all be in the Google Drive. Yeah. There's a Google Drive. It has every single program too, so I think you can actually spy on them and see what they're learning. But don't do that. I didn't advocate for it.
Hang on, I'll check it.
All right, we'll check it. So now you have this kind of method developed. So what's the next step? And I want to say something maybe controversial, but I'm going to say mass spectrometry signal is not inherently calibrated. If we look at these two chromatograms, these two peak areas, we might just instinctively want to say, oh, of course, the sample on the right is less abundant than the sample on the left. But have we validated that it's because biologically it's less present in the sample? There are a lot of reasons that the sample on the right might have a less abundant peak area than the sample on the left. Maybe I just didn't load as much protein on the column that time, or maybe, like Andy was saying, I left my tube out sitting on the bench longer and that peptide has degraded. Originally in the sample, maybe they were at the same level, but because I haven't maybe validated that this peptide is going to be stable over time, or that I've checked that I've loaded the same amount, or maybe digestion was slightly different, I haven't actually confirmed that these are quantitatively different values. All I can say is their peak areas are different. So that's why hopefully we should be motivated to calibrate and validate all of our data. So if you have been classically trained in analytical chemistry, a figure like this might look very familiar to you, where on the x-axis you might have samples of different quantities, different dilutions of a sample. And on the y-axis, we have whatever output quantity, whatever signal we get out of our analytical instrument. So for us, this might be intensity, like peak areas. And at a very high concentration, we expect to see a very high signal. At a very low concentration, we expect to see a very low signal. The portion of this where if I change the concentration, I get a proportional change in signal is the linear range. It's the dynamic range. And Andy mentioned this before, so if I dilute my sample by half, in the linear range, I should see my signal decrease by half. If I were to measure the same sample multiple times, I should get a reproducible signal from the same sample multiple times. So this is the idea of reproducibility, and I think this just kind of maybe inherently to us as researchers kind of feels correct. If I do the same thing multiple times, I expect to get the same results multiple times. And you might notice at the bottom of this linear range, there's some value where even though I'm changing the concentration, I'm not getting any change in the signal. This is because every analytical instrument everywhere, not just mass spectrometers, every analytical instrument everywhere has some value of noise, where even though I'm changing the signal at a very low concentration, I'm no longer getting a change, even though I'm changing the concentration, the amount that I'm putting in, I'm not getting a change in the signal because I'm measuring whatever the background noise is. So the point kind of down here, you can kind of think of it as a point at which we transition from the linear range and we start getting into the noise. That's a very important point called the limit of quantitation, the LLOQ, the lower limit of quantitation. We're going to be talking kind of about how to set up these experiments and measure the LOQ later. For now, we're going to focus on the rest of those points up at the top. We're going to focus on them because I think deep down we might think peptides from the same protein should give us the same signal, and that's not true. So one example, this is just some random protein that I detected in my yeast samples, and I'm showing the protein as though it's a linear sequence, so just the beginning of the protein to the end of the protein. All of the gray boxes are different tryptic peptides that I detected from it. So looking at this, I think, wow, I did a great job. That's some really good sequence coverage across that protein. If I took 2 of these peptides and I asked, what is the difference in their calibration curves? So if I made 2 calibration curves for each of these 2 peptides, and the scale on the x-axis is the same for both of them, going from 0 to 4,000 and 0 to 4,000. So each of these are the same dilution point. I hope that it's clear and you can notice that the level at which the linear range devolves into the noise is different for these 2 peptides, even though they came from the same protein. So what would be some of the implications of this if I were measuring this in some biological sample? If I used one peptide as a proxy to quantify this protein versus the other peptide as a proxy to quantify this protein. Why aren't they the same? If they came from the same protein, we might want to say, of course, they should give us the same signal and they should have the same kind of properties. And that's not true for a lot of reasons. One reason is we were talking about sample preparations and how I could digest it differently. Different peptides, even if they're from the same protein and under the same conditions, will digest differently. So the idea of digestion kinetics is one reason why peptides might have different properties in the mass spectrometer. Another reason is the chromatographic properties. Maybe a very hydrophobic peptide, eluting at the very end of my gradient, has very junky-looking chromatogram. Or maybe it elutes very early and it has an extremely sharp chromatogram. We talked about peak shape yesterday and how that affects our quant. Ionization efficiency is another reason we might have problems. So this is an experiment we had done several years ago where we took a protein and we got equimolar peptides from that protein. So digestion kinetics is not the answer here. We just took different peptides and we measured them by mass spectrometry and then we just ordered them. That's this rank down here. We had 40 peptides from this protein. We ordered them based on their mass spectrometry response, so their intensity. And some peptides fly very well from this protein and some peptides don't fly well at all. So not only are we dealing with digestion kinetics, but also their ionization efficiency or their flyability. Question?
Did you see patterns in composition?
We built a machine learning predictor. So this is Brian Searle from 2015, has a predictor called Prego that he tried to build a neural net that would predict based on some set of 11 physiochemical properties like amino acid length and hydrophobicity and blah, blah, blah. He tried to say, could I predict which one of the peptides from a protein would fly best? And it does not bad, but it doesn't do very well. So it's slightly better than if you were to randomly guess ragu, which is why he called it Prego. Slightly better than ragu. Prego is also an external tool in Skyline, so you can install Prego. But again, and he is the first to admit, it's not fantastic. So really the best way to tell is just empirically try. So I'm about to unload upon you a lot of people's various opinions for what experiments are important to do. And I think my best advice to you is to decide for yourself which one of these validations are best for your experiments. Like I said before, a clinical experiment that's actually going to be used for patient samples, maybe you want to be more rigorous. If you're doing, some biological perturbation experiment and you have maybe thousands and thousands of targets, say for your PhD thesis, maybe not all of these are appropriate for you. There's a couple really great kind of seminal papers on this topic that will tell you their recommendations. One of them is sitting in the room, so we can talk later after he leaves. No, I'm just kidding. So the Paradise Lost paper is a great one. That's Russ Grant and Andy Hoofnagel. And they talk a little bit about some of the experiments they wish they could see in more clinical chemistry studies. Another paper is by Steve Carr and every of his friends, where they recommended some best practices in what they call a fit-for-purpose approach. So I kind of refer to these as the Paradise Lost validations and the fit-for-purpose validations. We'll go a little bit more into depth. So this is the fit-for-purpose tiers of validation. Tier 1, which I feel is kind of demeaning to Tier 3, but Tier 1 is kind of clinical assays. And for this, Carr et al. Describe how they want labeled internal standards for every single peptide. They want reference standards in addition to these labeled internal standards. It should be extremely specific. It should be extremely precise. The accuracy should be defined. We've got kind of this middle one. Yes, Brendan. Not for clinical, yeah. Tier 2, I'll skip for now because it's just basically a compromise between Tier 1 and Tier 3. Tier 3, they say, is generally tens to hundreds of analytes and is used for exploratory statistics. There's basically no requirements at all for validations at this level. And then, like I said, Tier 2 is like this really weird in-between stage. So if you find these maybe not super clear for where your experiment would fall, I sympathize with you. So that is one set of standards that, while maybe you don't feel like you fit precisely into one of these bins, you can still look at a lot of these experiments, and they're really good experiments to do. Yes?
Just to explain a little bit here, so if you look at the list on the paper that this came from, there were like 100 some odd people, and all of these people were invited based on the variety of types of analyses they were doing at that time. So we had people that wanted to, like Andy, and people that wanted to make a diagnostic or clinical decision based on the outcome. And then we had academic researchers towards by standards for any of their stuff. And they, in order to stratify it and have some kind of recommendations for what others like to do and know where they fit in this kind of tiered strategy, the outlines were drawn up. And I'm sure there are many shades of gray in this but the point was that Tier 1 was not what the majority of people are doing and focused on very specific and small things. And at the other end of the spectrum, it is exploratory. It's going to be hard to generate statistics with people that have internal standards and things like these are a little more haphazard. And then the intermediate is trying to get whatever you're doing in Tier 3 eventually up through Tier 1 for in a way that things that you don't think are going to be beneficial for diagnostic purposes or for this kind of.
Yeah. That's an important point. You can start at a Tier 3, and if you start at Tier 3, you're not condemned to stay forever at Tier 3. You can start with a pool of potential candidates and then work your way up, and then tier 1 just do your candidates that have passed all these other stages of validation. So you can start with maybe not having internal standards for every single one of your candidates, and then start eliminating potential candidates that you don't have to buy so many internal standards. I would also say that I think this was kind of drawn up before DIA got really big, and the tens to hundreds of analytes was pretty much the limit of what you might be able to do with most targeted experiments, and now I think we're definitely pushing easily past the tens of thousands of analytes and doing fairly rigorous quant on them. So these will probably have to get redrawn once DIA becomes more mainstream. Yeah. Yeah. So I think probably my more preferred set of experiments is the Paradise Lost set of experiments. So these experiments are kind of laid out less as a set of tiers and more as a set of experiments, specific experiments to do. So this is where we were talking about things like peptide stability. So this would be a specific experiment where you would take standard peptides, spike into a digest, and measure the CV. So you might notice a lot of these are defined by the analysis of their coefficient of variation, and then it's different experimental setups to test how reproducible or how variable different perturbations to your system, to your assay will be. So we're going to be doing a lot of hands-on Skyline doing this, and I'll be showing you how you can just display CV right in Skyline to make a lot of these experiments super easy to do. I'll be going through today the experiments as they're described by CPTAC. So this is yet another set of recommended validations. And you can go to the CPTAC website, and a lot of the stuff I'm going to be showing you can be found on the CPTAC website. If you click on the About tab and you go to the right, there's like this guidance documentation for doing CPTAC style experiments that I think it's a little bit more into depth than the Paradise Lost or the fit for purpose descriptions. So in that documentation, they outline in detail 5 different experiments that you can do to test the validity of those peak areas. Remember, I was showing you, look at the super tall peak area and the super tiny peak area. Do we trust that quant? The top experiment is the response curve. We're going to be going way more into depth on that a little bit later. So we'll skip that one for now. Experiment 2 is repeatability. Experiment 3 is selectivity. Experiment 4 is stability. And experiment 5 is reproducible detection. So we're going to go a little bit through those, and I'm going to give you kind of the too long don't read of the documentation for each of them. Experiment 2, you would take the solution of peptides, your standards that you have. You would do 3 times the lower limit of quant, 50 times, and 80% of the ULOQ, the upper limit of quant, if your system has them. Upper limit, you have to have a saturation, so if you're maybe on an Orbitrap, you might not ever actually hit saturation on an Orbitrap. The idea here is we assessed pretty much the lowest thing we can quantify, something in the middle, and something at the very high end that we can quantify. This examines the inter and intra day reproducibility. If you do this on day 1, you get some CV. If you do it on day 2, you get some CV. And if you do it on day 3, you get some CV. And the reproducibility across the samples on each day and between each day gives you an estimate of how stable is this peptide going to be and how reproducible can I quantify it. So I think this is probably the easiest experiment that touches on all the different validations. So if you have any questions on this would be good.
Well, can you use heavy peptide?
Yeah, so this is kind of saying if you did this with peak area ratios. I've done this with label-free without any standards, and it worked just as well.
These are like low QC.
Exactly. If you come from a regulated environment, you might know this as low, mid, and high QC. Are there any other questions on this specific type of experiment? No? So experiment 3 takes 6 different examples of the matrix you might be measuring your analyte in. So if you're thinking about like maybe patient samples, maybe from patient to patient, the matrix may be changing. Maybe you have gender differences, or maybe you have like a particularly fatty subset and a particularly low fat subset, like lipid profiles. You would take those 6 matrices that might be interfering with your analyte. For each one of them, you would kind of do another one of those like QC style with a blank to see if there's any endogenous signal that might interfere with your analyte in that matrix. You would take kind of a low to mid level of analyte and check the signal of your analyte to the endogenous, and then a 50%, so like kind of a mid. And the idea here is that if the response of your peptide is changing in each of those 6 different matrices, that is not a particularly reproducible peptide to be measuring for some reason. Can you think of any reasons why different matrices might have different signal from your spiked-in peptide? I kind of gave the answer. I don't really do quick trick questions. 6 matrices, maybe they have different effects on the sample. Matrix effects. So matrix effects might be causing irreproducible quant values. So this is a test to make sure that that is not the case. For some experiments, this might not be quite as crucial to do if you're not working in patient samples. Like if you're doing a cell lysate or something, maybe you don't really expect the matrix to change too much between one batch of yeast and the next batch of yeast or something. But for other experiments, for example, if you're doing tissue blocks or something like that, where particular sections of the sample and homogenizing different sections of tissue would provide different matrices, this would be pretty important to do. So the next one is stability. This is freeze-thaw. So we talked a little bit about freeze-thaw experiments in the morning. So you would take, for example, a whole set of samples that you would prep all together and then split out into multiple aliquots. Some of them you would maybe place in your autosampler. This is, Andy had mentioned, in the McCoss lab, we do this test because it is so cheap and easy to do. Make 3 aliquots, place them in the autosampler, measure one of the aliquots on day 1 at time 0, one of the aliquots 24 hours later, another aliquot 72 hours later, just to assess whether or not the peptide is degrading or sticking to the sides of the tube. The freeze-thaw test as well, even though you might think that freezing should have no effect on the peptide, we've actually found that it does. So assessing this, make sure that if you have some standard, you're going to leave it in the freezer for some longitudinal study, that you're not going to have your quantitative values affected. Question?
So sometimes the blood is collected and it's frozen, and then plasma is disconnected plasma if you want. So that's how our labs store plasma samples. So this one, for example, if you have such plasma, does it make sense to conduct this kind of experiment? Like slowly, let it stand for 6 or 7 hours. So
let me just double check that I understand the system. So you get a set of plasma samples.
Yeah, so ideally maybe you should have conducted experiment when subject's blood has been set at room temperature for 6 hours, then frozen, then the plasma was extracted, right? Rather than plasma. So can you do this experiment especially from plasma? Does that make sense?
Oh, you certainly could. Yeah. So there was a similar experiment done on breast tumor samples, I believe, where they either harvested and then froze immediately, or harvested and waited a bit and then froze. And I think kind of the take-home message is, if you have particular targets or analytes that do degrade very quickly, you wouldn't measure those. You would try to find a different target that's not degraded. If it's already happened with the samples, it might be a good test to do if you had a reference material, like some reference plasma sample, to do that test and see if any of your targets are affected that way. If it's already how you have the sample, you gotta do what you gotta do, right? Does anybody do similar experiments in a slightly different way? This is just the CPTAC guideline, but it probably for me was the one that made the most sense because I was already kind of doing freeze-thaw tests like this anyway. So one thing that I would add to the CPTAC guideline, that's something we learned firsthand through a student making a tragic mistake, was 4C and -70, -80 is good, but if you ever use a -20, that might be another one to check. So we found pretty extensive degradation of some target peptides in another student's assay when she unintentionally stored it in a -20 instead of a -70. So if you do use that kind of storage system, that might be another good one to check. So the last set of experiments, to give you a feel for the different experiments before we look at real data, is to do what is referred to in my lab at least as the 5x5. The 5x5 is doing 5 samples each on 5 different days. So you end up with 25 total samples, but this idea is just kind of to go one step beyond that mini experiment 2 where we did the 3x5 kind of quick test. This is the 5x5 thorough investigation of inter and intra-day reproducibility. So these are, you would start from an actual sample and do your entire sample preparation protocol from sample 5 times, 5 different samples, and then you would do it again the next day, and the next day, and the next day. And the idea here is that any inter or intra-day variability in your sample prep and your entire system, your mass spectrometer, your targets, your stability should be captured in this kind of study. So if you do a 5x5, you should already have your protocol set. If you change your protocol, that might change the type of data captured in a 5x5, so you would then have to do it again.
Could sample 1 in that case be a pool, or should it just become a single individual, or does it matter?
I've always done it with a pool. I've never done it with different patients on different days.
No, I just meant like sample 1 being from one individual, or could it be a pool for multiple individuals just to have enough volume?
Oh, I see what you're saying. Yeah. Yeah.
But in both cases, they're both pools from multiple individuals.
Yeah. When I do this, I'm doing this in yeast samples, so it's very easy for me to just grow a liter of yeast and have pellets for days. So that was a very quick overview of the different experiments, and I didn't want to kind of belabor the point because you're all educated individuals and can read those papers yourself. One point I do want to impress is that assay development and validation should eliminate candidate targets. So this is a target list from Clark Henderson, a great scientist in Andy Hoofnagel's lab. He did the vitamin D binding protein assay. So we'll be working with that data to look at some of what the data looks like for these different types of experiments. But he started from, for example, that vitamin D FASTA, he had 48 peptides under the digest conditions, the peptide setting conditions that he set. Out of the 48, a lot of those had either less than 6 or more than 25 amino acids. So that automatically eliminated a lot of candidates because he wanted tryptic peptides that fit this amino acid length. He also culled about a dozen peptides because because they did not have, to him, ideal amino acids. He eliminated one of those ideal candidates because it did not have good precision. It had less, it had over 20% CV. He did a stability test, which further eliminated 3 of the targets. They weren't stable under freeze-thaw conditions. 2 more targets were eliminated because they didn't have a good signal intensity in plasma. And then finally, he ended up with 7 targets that had no interferences, good signal intensity, stable to freeze-thaw, had good precision, blah, 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 blah. So I like this figure because I think this is a good message for us to remember, is that he started with almost 50 candidates and only 7 of them survived all these validations. And that's not a bad thing, that's a good thing, because now we know these 7 are good proxies for the protein. So it's not a more is more, it's about quality, not quantity of your analytes. So for the hands-on for this section, we're going to be using the data that Clark collected for these different types of experiments. This is a great read. It's the vitamin D binding protein paper that came out a couple years ago. This was touched on by Andy, but one of the important conclusions that they made from this paper was that immunoassay had suggested that vitamin D binding protein concentrations varied by race, and they found that it didn't look like that was the case based on these validated targets. Their different experiments are labeled maybe a little differently than the CPTAC guidelines, but it gets at all of these same questions. It assesses all of the same validations. This data is in your flash drive under Henderson serum validations, I think it's called. These are all the data that they included with the manuscript. So this is a completely transparent experiment. You can look at all of their data and validate the targets that they said were validated. They're all Skyline files. Each one of the different tests that they did is a Skyline file. I would encourage you to not spend all of your time in this hands-on section doing just one of the data sets, but try to look at the different data sets. They are kind of set up the same way, but the data is very different because it was different experiments. I don't know if I have Skyline on this computer, but if I do, I will try to open it up and show you some of the tricks that I like to use. Here we go. So I'm just going to pick out, ooh, which one do I want to pick out? I'll pick out stability. And I'll pick freeze-thaw stability. You don't have to follow me. But I'm just going to, oh no, that's okay. I'm just going to show you some of the little Skyline tips and tricks that I like to use when I'm analyzing this type of data. So once again, just to orient you to the Skyline document, he's got 1, 2, 3, 4, 5, 6 different targets over here. This opened up for me. It might be a little different for you. This opened up for me with detection times on the bottom, areas on the top, and the chromatogram pane in the window. I personally like having my peak areas on the bottom here, so hopefully you're pretty comfortable with rearranging panes, so you're more than welcome to rearrange them however you prefer. One of the things that we were just learning is how to group the data. So we were doing kind of the group by sample group thing. So one of the things I like to do right off the bat is open up my results grid. Oops, sorry, it might be document grid. And go to my views and go to replicates. So these file names here maybe aren't informative to us, but because this was published with extremely high transparency, if you go in this data drive, there's a folder here with a PDF that has full descriptions of all of the data files that are in these Skyline documents. So this is typically a place I like to start when I'm opening up an experiment and I've just pulled in all the data. I like to organize it into sample groups or rename the file names to something more English language legible. So I had opened the bias file. This trackpad is not super great. So I opened freeze thaw. The mouse doesn't work. So I might want to rename some of these files. Oop, you're okay. I might want to rename some of these files to match something that's a little bit more intuitive. So maybe I would rename these fresh, freeze, 1, freeze, 2. So I think it went, oop, yes. Yeah, I opened the, oh, this trackpad. Oh, I can do it. Oh, hello. Yeah, so I'm in, oh, I'm not in bias, sorry. I'm in stability, and I just opened the freeze thaw stability file. And this is just to show you. I encourage you to open up different files. It especially is nice if you open up different files than what your neighbors have open, and then you can share some of the things that you're finding in the different files. So I might want to add to this view. Oh, sorry, I'm not used to this trackpad. I can do it though. I'm going to go to my document settings, and I'm going to add an annotation for sample group. So I want something called sample group, and I want to make it on my replicates. I think you might have already done this. Did you not already do this? If you haven't already done this, I'm just in document settings, so up on settings, I went to document settings. That opened up this document settings pane. And now I'm just setting up some annotations so that I can group things to explore the data a little bit easier. I'm going to choose this as replicates. This applies to replicates. Now I have a sample group annotation, and after I've hit OK, I should have this pop up if you didn't already have it there. So sample group, I think it was fresh, freeze 1. I think it was something like that. So now if I right click on my peak area pane, I should have group by, and now I should have this option for sample group. Did anybody want me to do that again? Are you, yes, was that a yes? Yes, that's again. So I had gone, one more time, settings, document settings, annotations. For me on this Skyline, on this computer, I didn't already have a sample group. So I made one by going add. And then I can call this whatever I want. And then I chose replicates because I want this annotation for each of my files, so each of my replicates. Then when I hit OK, I had this guy show up in my little annotations. Maybe you have a ton more in your annotations pane. I think you might. I checked it. I hit OK. I hit OK. So now I would right-click in my peak area pane. Your peak area pane might still be on top of your retention time pane just because I prefer, I don't know, my eye just likes seeing the retention time in the top corner and the peak area in the bottom corner. If I go to group by now, I should have an option under the group by for sample group or whatever I just called that annotation that I just filled in. So now I have those two replicates of fresh, the two replicates of freeze 1 and the two replicates of freeze 2. And then I just wanted to show you how you can check the CV values box. And now it is showing me CVs instead of showing me peak areas. So what changed here was the y-axis. Instead of showing peak area now it's showing peak area CV as a percentage. It's still split in light and heavy, so maybe I need to normalize to my heavy. Normalizing, you right-click in that box, go to normalize to heavy. So now I'm showing peak areas normalized to the heavy. So all of these peak areas normalized to heavy. We're under 7%. It's just a trick that I like to see these. Oh, sorry, I was on the protein level just then. So it looks like this freeze, one of the freezes, looked like it didn't have great stability. Freeze 1, all of these had stability under 10%. Under 4%, that was a good one. Oh, it's maybe not picked right. This looks like it might be this. Whoa. So that's just one feature that can help you analyze the data. There's a lot of files in here. Just to see what this type of data looks like. See, they're the same targets. They did all of these validations. Maybe you want to look at linearity. We can pick one of these. Linearity. You don't have to follow me. I'm just opening up a different file to show you that it's different looking data. So this data, right off the bat, visually looks very different because it was a different experiment. All of these different replicates. Maybe I would go back to that sample key and look at what these are. Maybe I would do the exact same thing, fill in some annotations. Yeah.
How do you get your replicate comparison?
Yours is a what? A minus?
Lines.
Okay. Well, I will be coming around to help you out. Like I had said, sometimes this works particularly well if you and your neighbors are looking at different types of data, and then you can kind of compare notes. But we got some time to play with these.